Hello everyone and welcome to my poster. So I'll be presenting my method MODEST, which I'm currently developing and which stands for Modeling of Epigenomics and Spatial Transcriptomics data. So I will give a brief introduction about the spatial transcriptomics and single cell multiome data. So the spatial transcriptomics data is able to capture the gene expression along with its spatial location in the tissue, whereas the single cell multiome data is able to capture various molecular information, but specifically it is capturing gene expression and chromatin accessibility information at a single cell resolution. Spatial transcriptomics data is not able to capture the chromatin accessibility information in a spatial manner. It's just only able to capture one type of information, which is the gene expression data. So we are integrating the single cell multiome data with the spatial transcriptomics information so that we are able to predict the chromatin accessibility information for each of the spots in the spatial transcriptomic dataset. To do so, we do have certain challenges, and the first major challenge that we have is that there are batch effects between the spatial transcriptomics and the single cell RNA-seq dataset. And our second challenge is the alignment alignment of the single cell RNA-seq and single cell ATAC-seq data that our model will try to do for this for the single cell at a single cell resolution. So we are using two different types of variational autoencoders in our model, and we are using variational autoencoders because they are able to perform dimension reduction, which is done by the encoder, and then once we obtain the embeddings due to the dimension reduction, the decoder uses that information and tries to generate an output which is very similar to the input. And it does do so by capturing the distribution of the embeddings. So we have the spatial transcript. So we have the gene expression from the spatial transcriptomics data and the gene information and the gene expression information from the single cell multiome data, which are used as input in a variational autoencoder 1. And for a variational autoencoder 2, we have the single cell ATAC-seq data, which is used as the input. And once we get the embeddings for the single cell RNA-seq and the single cell ATAC-seq, we use contrastive loss to align them together in a shared latent space. So once this model is trained, then we use the decoder of the variational autoencoder 2 to perform prediction of the chromatin accessibility and prediction of chromatin accessibility for the spatial transcriptomics dataset. So these are the loss functions that I'm currently using for the MODEST method, and there are some key factors that I'll just go through for the loss function. So to capture to remove the batch effects, we are calculating a separate KL divergence for the spatial data set and for the single cell RNA-seq data. And for the VA2, we are adding the contrastive loss so that we are able to align the RNA-seq and the ATAC-seq in a similar fashion. And we do have the structure loss as well to maintain the structure, the integrity of the matrices. So for the simulation, we currently do not have a single cell ATAC-seq or the single cell ATAC-seq, we do not have the chromatin accessibility information for the spatial data. So since we just have the gene expression data, we are splitting the data set into train and test, based on the genes. So there would be some amount of genes in the trained data set and some amount of genes in the test data set. And the assumption is that the test data set would be used as the ATAC-seq data set or the chromatin accessibility data set, and the trained data set would act as the RNA-seq or the gene expression data set. So we did the simulation. We did check our method with different train test split ratios to assess our model's performance. So as you can see, we compared our model's performance with PCA, CCA, and tangram. So if you look at the line plot, which is plotting the mean with a 95% confidence interval for different split ratios. So when you see the 60-40 split, it means there's 60% of the data set in the test data set and 40% of the data set in the trained data set. So we do split that, and we see that the tangram method is performing much better than the other PCA, CCA, and our modest method is performing much better than PCA, CCA, and tangram. And in the other plot, we do notice that modest is performing better than PCA and CCA, and though the median is similar, though the median of CCA is similar to the median of modest, that we have many negative Pearson correlation coefficient values for a lot of the genes. So in the future, we would want to predict the chromatin accessibility information on Alzheimer's transgenic and wild mouse models, and also gain a deeper understanding of the transcription factors activity, and also on the enhances effect on chromatin accessibility in a more spatial manner. Thank you.